and translation? We love this question because it's giving us information. Maybe we already know it, maybe we don't, maybe it's reminding us, but we talked about using this and coming back to it, right? So if you won't fail state testing, so you get like a number 1 through 5 to score, and then you collectively through all of your different end of course exams, so like you'll have it for English and language arts, you'll have it for math, you'll have it for history, you'll have it for government, biology, I think algebra 1 and algebra 2. You have several tests, okay? And you won't fail. You'll get a score on those 7, 8 tests that you take from your 9th grade year to your 12th grade year. And you're building a score, okay? So you get, like, if I don't remember if it's 7 or 8 tests, but let's say it's 8. So you would have a possibility of getting 5 points for each of those tests. So you could potentially have like 40 points if you got 5s on every single test. That's a pretty high expectation, right? So we said this testing series is saying basically if you're better at math than at biology or English and language arts, maybe you'll get 5s on your math tests. Maybe you'll only get 2s or 3s on your biology and ELA, but what you have to do is get a cumulative score of a certain amount. I don't remember what that point number is, but there's like, you must hit this, at least this number of points to graduate. All right. And that doesn't matter how you score on what of any of those tests, but it's just collectively, you need to have this number. You must receive this number. That's all right, Tabby. All right. So that's kind of how it works. 18 points. So if you fall short, we're going to just keep testing you. All right. There are some alternate paths that you can do. And so if you have another teacher, a special education teacher, or if you have an advisor, those would be teachers to talk to, and they will talk to you about different pathways around that. There's always pathways around it, but we'll just keep testing it. So say you take the biology test this year and you don't do super well. Okay, you get maybe a 2 or a 1. You can get another chance. Oh, hey, Miley, can you grab me my charger out of that bag over there? My computer's dying. You can test again. All right. You get another opportunity. So you don't just get one run at it. You can take these tests multiple times. Obviously, no, like, tell me how many people love to go take tests. Do you love to go take tests? Right? I don't love to go take tests. So if you don't love to go take tests, I say you study really hard and do what we're doing now to be successful. And then, you know, hopefully you won't have to go back and take it again. But it is a nice opportunity that it's not like a do or die, you know? It's not a do or die. So that's the cool thing. So hopefully that helps you a little bit. Is this some of your first tests that you've taken? I'm guessing this might. Nope, it's not a one-time thing. You can definitely. Now, I would say your best shot at getting your best score for biology is going to be right now. Okay? Because if you take it again next year, will you be in biology class? As long as you pass biology, you won't be in biology class next year. So you can take the test again, but will this be as fresh on your mind? Probably not. Okay. So yeah, so we want to just do really, really well this year. We want to show up, we want to give it our best, and I'm going to help you do that. All right. And again, Mrs. Sheets is holding those help sessions every single week that are just state test focused too. So go attend those. That's a huge benefit. Okay. It's all right. You've got this. I'm telling you, half or more of taking the test is the strategy, not the content. Right? Half of it is the strategy, not the content. And so, Jesse, you're going to be able to go in and you're going to be able to do what we did yesterday and done this week. We're going to look for those keywords. We're going to use the diagrams that they give us to help us remember what we don't know. And we're going to make the best guess we can on some of these answers, right? By just getting rid of nonsense. Okay? You've got this. And we're just going to keep preparing and preparing to help us get through it. So yesterday we worked on this question last. And it was about how water molecules move in and out of the cell. All right. We call this diffusion. Does diffusion require energy? Does diffusion require energy? You want to say hi to everyone, Ren? No. Nope, it does not require energy. Diffusion is a energy-less process. Remember how I said if I crammed all 50 of us in a small room and then I opened up the door and let some people go to another room, would you stay in the crowded room or would you move to space? All of you said, I would move to space. Nobody wants to be cramped up, elbow to elbow with smelly people. Nope. Osmosis is a form of diffusion, right? It's a form of diffusion. It's water. So it doesn't require energy either. All right. These are what we talked about earlier this year. Again, the types of solution. Isotonic solution means that there's the same amount of solute outside of the cell as inside the cell. And solute is kind of the same as just a fancy word for stuff, right? Or salt, or the water is following the molecules that are in the substance. So when there's the same amount as the outside as in the inside of the cell, our molecules are going to move out and in at the same time. In a hyper, we talked about when you're hyper, right? Like your energy is going out of you. And it's the same way in a hypertonic solution. Water is leaving the cell. Just like when you're have all that energy and you're hyper, the energy leaves the cell, right? And when the water leaves the cell, it kind of shrinks up. So if you see a shriveled looking cell, that's an identifier of a hypertonic because water has left the cell. And then we talked about hypo or hippo, right? What do you think about a big old hippo? A big old round hippo is when the water comes into the cell from outside. So that means there's more solute inside the cell and water is drawn inside the cell and it makes the cell all big and fluffy. All right. It's a good old bloated cell. Think about your belly when you drink a ton of water, right? You feel like, whoa. So that is something. If you want to screenshot this for your notes, this would be a good notes section. Isotonic means it's going in and out at the same time. Hyper is it's going out. Hypo is coming in. Yep, snipperoo. Go right ahead. And there's another page you can snip if you like these diagrams better, right? When a cell gets so much water in it that it can't handle anymore, it can bust, just like a water balloon. All right, we called that lysed. Lysed.
Can I add something?
Sure.
So when I think of like hypotonic, like when I saw that image of the cell like getting bigger, it's like when I realized this, I imagine it as follows. So basically, it's basically like if you drink, like if you drink so much water at like gallons of it at once. It basically is like the same thing, except it occurs nearly everywhere and even in your brain. So I was wondering if something like that is kind of related to hypotonic stuff, right? Because that's what I think of every time I see it.
Um,
In other words, the cells, like not just in your muscles but in your brain, that get big because they absorb the water because there's like a lot of it, and you can't handle it and all that. I was just wondering that.
um, it's going to depend on where your cell is at, right? So your body has a lot of regulators. So your brain cells are probably not going to get hypotonic and lysed and popped to that kind of extreme. Because your body is always fighting against that, right? Too much water, yeah, just like what Sean's saying, too much water, too much of anything is a bad thing. I mean, you can drown yourself in water, right? It's not always a good thing. So everything in moderation, but your body is going to constantly fight that, right? Because your body is fighting to stay in homeostasis. Now, when you isolate cells and put them like in petri dishes or beakers or whatever else, that's when you can, a little bit more experimental. You'll see some of those things on a cellular level versus your body is a giant multicellular organism. So when you start flooding your body with water, it's going to start counterbalancing that a little bit on its own. All right. Good question. So again, diffusion is when molecules move from a high concentration to a low concentration. All right, facilitated diffusion. Remember, it just means facilitated means help. Diffusion with help, that means it's still diffusing, but these molecules, instead of just being able to permeate through that cell membrane, they actually need a channel. They actually need a doorway to let them through. All right. And then this is a facilitated diffusion with a carrier protein. I kind of like to think about this like as airport security, right? So like there are people that have already been pre-checked by TSA, and they can walk in and they can go right through security because they've already had pre-screening. All right, this situation is normal security, right? You're going through, you're getting checked by the radar. Are you what you say you are? Do we want you in the cell? And it comes through that way. This situation over here on the far right is kind of like you got through and then they saw something on, on you or your bag that they didn't know what it was. So they send you to this other line and they check you and your stuff a little bit more securely. All right. So that's kind of like cell security, just like the airport. All right. So this is another example question, right? These are some of the things that you could be asked to do on the test. They're not just A, B, C, D questions, they're application questions. So let's practice this. All right. Is there someone who would like to read the question? It's okay, Jake. It happens. We're glad you're here. All right, Pippa is going to start reading this question for us, and then we will start dissecting it.
Alrighty, goodness, I forgot to click on the mic. Anyways, I gotta zoom in just so I can see it. A student fills a beaker with a 5% salt solution. The student places a dialysis bag containing a 15% salt solution into the beaker. Then the student measures the salinity of the salt solution in the beaker after 5 minutes. The dialysis tubing is permeable to salt and water. A. Place a label in the blank box to show the net direction, I almost said destruction, in which water moves immediately after placing the dialysis tubing in the beaker. B. Place a label in the blank box to show the net direction in which the salt moves immediately after placing the dialysis tubing in the beaker. Could I read the stuff on the right as well, if you don't mind? Oh yeah, you're muted, but okay. So the answer choices are as follows: salt into tubing, salt out of tubing, water into tubing, water out of the tubing, no salt movement, no water movement, and the experimental setup goes as follows: 5% salt solution, dialysis tubing, 15% salt solution, A, water movement, and the B is for salt movement. Thank you.
Good. All right, so let's just look at question A. All right, let's look at question A. This is a lot. Anytime there's a big question like this, I'm going to just break it down, right? Let's break it down. What is the very first question that they're asking us to do? First question. Put it in chat. Say it on mic. What's the first question? Yes. Good.
Oh, sorry.
No, you're fine. So it's asking you, Sean says it's asking how the water will diffuse, right? So it says place a label on the blank box to show the net direction. So it says A, the net direction in which water moves after placing the tube in the water. So we've just placed this tube in the water. How will the water move? Well, we need to look at where the greatest amount of stuff or solute is. So the solution in the beaker is 5% salt or stuff. And inside this dialysis tubing, it is 15% salt or stuff. Okay. Which way would the water go? Diffusion and osmosis, it moves from what to what? Tell me in chat. In diffusion, what are it's going to move high to low. Good. So where is the water going to move? Is it going to move from the salt solution on the inside? Is it going to move out? Or is it going to move from the salt solution into the bag? Where is the more stuff that we're trying to balance out here? Yeah. Yeah. So the water is going to move into the tubing. So what you would do is take it and drag that in there, right? Because there's a higher percent of salt in this tubing. So what we're trying to do is even it out so that there's an equalness of water per solute or water per stuff. So it's going to move into, the water is going to move into the dialysis tubing. What about the salt? How will the salt move? So the salt is in this bag. It's 15% here. It's 5% here. What is that bag going to do? Yeah. So some of that salt is going to say, there's more space out there, guys. I see a lot more space outside of this tubing than inside of this tubing. So the salt will move out and the water will move in until it hits equilibrium. Does that make sense? Until there's the same proportion on both sides. Salt out of tubing. We go here. Does that make sense? Give me a smiley face. So the salt is going to say, there's so much salt in here and I'm looking out there and there's so much space. So they will move out. And the water from the inside is saying, they need my help balancing out that. That is very concentrated. It's dehydrated in there, right? It needs water. So it moves in and out. Good. Perfect. A little bit of confusion. Don't let it confuse you. All right. Remember, for diffusion, salt is going to move out. Water is going to move in because it's trying to hit equilibrium. I just want you to really keep that balance of equilibrium in your head. If I'm trying to have equal parts water and equal parts salt, on both sides, how are things going to move around to make that happen? Does that help? To get it balanced, water's going to go in and some salt is going to go out, and then we're going to get to this spot where we call isotonic, which means it's the same on the outside and on the inside. It's at equilibrium. All right. Hopefully that helps some. Let's do another one of these questions. I think that will help us. All right. Would anyone else like to read? Any other readers? All right, Pip, go ahead and read this for us.
Alrighty. The diagram shows an incomplete cross-section of a cell membrane. 3 different particles are shown moving across the membrane into or out of the cell. The key goes as follows. The blue circles is O2, which I assume is probably oxygen. The red triangles is the glucose, and the yellow squares is Na, which I assume it's a type of salt. I don't know. But anyways, determine the appropriate transport mechanism of each particle and move the correct membrane segment into each blank box to complete the cross-section diagram. Move only one membrane section into each box, and the picture as follows. Outside the cell, and then below that, inside the cell, ATP pump, a channel, and phospholipids. Thank you.
Good. So down here, these are our answer choices. All right. It's going to be a drag and drop to these different boxes. So this is outside of the cell. This is the inside of the cell. All right. And these are our 3 choices. Now we also see the direction that they're moving. Some of them are moving in. Some of them are moving out. Let's look down here. What if one of these is much different than the other? Which one of these? Let's just say this is A, B, and C. Yes. You're right, Ava. Good job, Aria. Yes. A is different than all the others because it has... Why is this not working? A is different because it has what? Yes, it has ATP. What does that tell us? What kind of transport is that when we have to use energy? Good. Is it natural? If we have to use energy, is it like a natural desire of that molecule? Is it a natural movement? Or is it like ATP equals force, right? It's pushing it out. So if we look at these, let's look at these and you tell me if they're moving from low to high or high to low. Let's look at these one at a time. This one right here, is it high to low concentration? How are the molecules moving? Or from low to high? How are those molecules moving? High to low. Do we need energy to do that, or is that what molecules want? That's what molecules want. Yep, you're right. You're right. We don't need energy for this. This is natural. That's what they want. They want to move from areas that are cramped and tight to areas where they have space. So we don't think it's that one. Let's go down here. The blue dots represent oxygen. All right. They're coming through the membrane. How are they moving? From high to low or low to high? Tell me right here what's happening. High to low or low to high? Good. High to low. All right. Again, we just said that's favorable. That's how molecules want to move. They want to move towards space. So that doesn't look like it. So without even analyzing that last choice, we're going to assume that's probably where we need to put A, because we haven't, these two we know don't need energy. But let's just look. What's happening down here? High to low, or it's moving this way, sorry. High to low or low to high? Yes. It's taking molecules that have all this space and room. It's like you have a room to yourself, and it's saying, never mind, you don't get a room to yourself. You get 50 roommates. Is that desirable? From having your own room to having 50 roommates. Right? If you had the choice, you'd be like, no way. And so in order to do that, that's a kind of like a forceful situation. We need energy to make ourselves do that. So we need to move A right here to our ATP pump. Okay. Now let's look over here at the oxygen and the glucose. How do we know which one, how do we know which one is which? Which one needs the channel and which one doesn't? Yes. Which one? So oxygen and glucose. We need them both, right? What do you think? Glucose is bigger. Agreed. We go back to this channel right here, right? So this one I feel like would be a trickier guess if you weren't sure. It's going to be the oxygen is going to go through without a channel. All right. The oxygen does not need a channel to go into the cell. Yep, the glucose has just kind of a different shape. So it's going to use a channel to go in and out of the cell. It's going to use a door, right? Good. Good. Let's keep moving so we can get a couple more questions. Are you feeling better about movement in the cell? Are you feeling better about movement in the cell? There we go. A lot more happy faces. All right. Again, and I don't want it to be like I'm going to be able to ace every single one of these questions. They all make 100% sense. They probably all won't. But if you have a solid foundation about how things move in a cell to get to equilibrium, what is natural and what is not natural, you're going to be able to solve most of these situations. Okay. All right, let me read the, or Ari is going to read this. Okay. Ari, you go ahead and read this one for us.
Okay. 3 identical red blood cells in side view are placed in 3 different saltwater solutions as shown in the diagram. Predict the net flow of water, H2O, into or out of each cell. Then predict the effects of this water movement on the structure of each cell. Assume the volume pictured inside the red blood cell is the same as the volume pictured outside the cell. Do you want me to read the bottom 2? Okay. Place the correct picture in each blank box to show the net flow of water into or out of each cell, and then place the correct picture in each blank box to show what will happen to the structure of each water cell after the water movement occurs.
Perfect. All right. So let's do this. All right. Up here at the top, we're just going to look at the picture first. Up here at the top, we see the black dot is salt. The blue dot is water. Okay. And let's just break this down. Let's break it down. In this very first picture, what do you see inside and outside of the cell? Does there look like there's more salt outside or inside? Is there more salt outside or inside? There is more salt outside. So where is water? How is water going to move to try to create equilibrium? Water is going to go to where the stuff is. Okay, right? So water is going to move to wherever the more stuff is. So if there's more salt in the cell, water is going to go in. If there's more stuff on the outside of the cell, water is going to go out. So if we're saying that there's more salt on the outside of the cell than in, then water will move out. Okay? So we would drag this where it's showing that water is leaving the cell. Whoops, I went too far. Does that make sense? Water is leaving the cell because there's more salt out there and it needs to balance out this water to salt ratio. So if water leaves the cell, what happens to the shape of our cell? What happens to the shape of our cell? Is it going to burst? Is it going to be the same? No effect. Or is it going to shrivel? Yeah, good. It's going to shrivel. Awesome. Guess what? That one is done. So it's like, let's just block that sucker out of our minds, right? Moving to this one. Let's look at it. Where is more salt? There's one salt molecule. Is there more salt on the inside or outside of the cell? Everyone is saying inside. So if there's more stuff on the inside, what's water going to do to try to balance it? Is water going to go outside, inside, or is it going to go in and out at the same rate? Are we already at equilibrium? You're right. The water is going to move in. All right. There's a lot of salt in there. Why did my pencil get so big? We're going to move it in there, right? To try to balance things out. So if water moves into the cell, what happens to the cell? Is it going to stay the same? Or is it going to get big, 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 and then potentially, yep, it's going to burst. It's going to lyse. Okay? So this last one leaves us with very few choices. But if we look, it's about the same amount of salt outside as inside the cell. So would I sit there and count the dots? Would I suggest you count the dots outside and count the dots inside to see if they're the same? You're right. No, don't do that. Okay. If it makes sense that the only answer choice is that the water moves in and out at an equal rate, and you're looking at that diagram and it looks about that way, then trust your instincts that you are right on the other answers, okay? And move that sucker there. And if things are moving out and in at the same rate, just like a rotating door, things are coming in and going out at the same rate, then what is that cell going to look like? Does it make sense that it would have no effect? It does. Okay. Awesome. Give me your feelings. Give me your thoughts. How are we feeling? Things move in and out of cells, how substances move, and how water moves. Right? We're talking about diffusion. Diffusion is of molecules. Osmosis is the movement of water. And on the go, I see a couple meh and sad faces. Please tell me what you're struggling with so I can help answer those questions. Zero, I'm not mad. I just want to help you. So tell me where your questioning or confusion is. I just always want you to think about equilibrium. What is happening in that environment to hit equilibrium? Molecules are moving and water is moving. They're moving to hit equilibrium. Okay. Sure. I like these diagrams though, because these right here are showing you pretty specifically, right? They're labeling it for you. They're showing the arrows and they're telling you what's on each arrow. And they're not even showing you the shape of the cell. They're telling you what it's doing. It's showing that it's bursting. You don't have to guess that that's bursting. So water moves to where solute is to maintain equal. Absolutely, Faith. Yep. Water is going to move to wherever that solute is. It's like they're all kind of just playing a role in maintenance, equilibrium maintenance. Everybody is doing their part. The solute, it wants to go from high to low, right? It wants to get to a spacious environment. It wants its own space, its own room. And the water is trying to help it. It's trying to create space for the molecules so that really everything is happy, right? When we're at equilibrium. So all parts are working to try to hit equilibrium. Yeah, I'm so glad that helps. Let me look here at the questions we have left, because I would like maybe to move to, are you okay if we move away from these solutions for a minute? So I feel like we've done a lot of these today. I do have a couple more like that, but I think we could hit up something different. Let's do this question. Guys, I'm not going to lie to you. If I saw this on the test, these are my worst nightmare questions. Okay? There are like 3 different things going on here, right? There looks like there's some kind of an experiment. There's charts and there's graphs. And I'm just like, for me personally, that is like visually overstimulating. Does anyone else feel that way? I mean, honestly, that is just so much. I'm just like, come on. Yes. All right. So the thing I love about doing your tests online is when I was in school and we did these on paper, I could literally cover it up with my scrap paper and look at it piece at a time. But what I just really want you to do, guys, I want you to just hone in to the very first section. Okay? Like legitimately, pretend the rest of it isn't there. You just have to tell your brain, all right, this is not 1 question. This is like 3 questions. So we're going to go in and we're going to look at one part of this and break it apart, right? And you may need to do this. You may be sitting there. Everybody take a deep breath. I'm not lying. I'm not trying to be whatever. You may need to just take a deep breath, right? And be like, all right, I can do this. Everybody tell yourself, I can do this. I can do this. Because you can. All right, we can do this. So I'm going to read this to you. It says the following question has 2 parts. And we're like, well, duh, look at all of this. First, answer part A, then part B. So we're looking just at part A, even just that top part. Students investigate how light intensity, all right? So I'm look at the diagram as we go. Okay, I see the light. And I see the ruler underneath it. All right. Light intensity affects the rate of photosynthesis in an aquatic plant. I want you to tell yourself what these things are. What is photosynthesis? You're like, oh, sweet, I know that word. We talked about that in science. Photosynthesis is how a plant makes what? Good. Energy. Energy from light. So we're like, all right, this diagram's just showing me the light, the plant, and how it's making energy. So we're looking right here at that diagram, and it's like, okay, there's a little test tube. It's an aquatic plant. If you don't know what aquatic is, you can guess by looking over here. The blue is water, so it's under water. So it's a water plant. We're looking at where the light is. It's a certain, they're measuring the distance of the light from the plant. I'm figuring this out. So the students place a light at different distances from the plant and record the number of bubbles produced. All right, so I'm kind of starting to see this experiment come to life in my mind, right? So I'm thinking, all right, so these kids are moving this light back and forth from plants and measuring like how much of the oxygen is being put into the water in these little bubbles to tell me how much energy it's making, remember? Because in photosynthesis, what is an output of photosynthesis? What do plants give us that we need to make energy? Oxygen. Yeah. All right. So let's just move down. We haven't even looked at the question yet. We're just making sense of all this craziness. All right. So we've got a light that's moving. We're looking at the bubbles of oxygen that are coming off aquatic plants. All right. Now we're going to look down here. They're moving at different distances. So we're seeing that it's going from closest to farthest away from the plant. And now we're looking at the amount of bubbles. What's happening as the light gets further away? Is there more oxygen or less oxygen as the light moves further away? Yeah, there's less oxygen. So as this light moves this way, we're seeing that the plants are producing less oxygen bubbles, which means ultimately less what? Energy, right? We can't measure the energy, but we can watch those oxygen bubbles. We can see and measure that. So what inference is supported by the data? You've already answered that question, right? How cool is this? You've already answered that because you've looked at this and made sense of it. So I want you to tell me, which one of these is it? The plant produced more oxygen as the light intensity increased, right? So light intensity is closer, the more oxygen was created. B, the plant takes in more water when the light intensity decreases. The plant requires more energy for photosynthesis at lower light intensities. Is that what they measured? Did they measure the energy with the light intensity? Did they measure the water with the light intensity? No. The plant needs less carbon dioxide for photosynthesis at higher light intensities. Were they measuring carbon dioxide with the light intensity? All right, look, guys, these are oxygen bubbles. How do we know they're oxygen bubbles? Because we know that green plants produce oxygen that we need, right? Green plants produce oxygen. So we know that these bubbles that are going up are oxygen bubbles. So all we have to do is go in here and say, all right, this question is saying it's oxygen. It's comparing oxygen and light intensity. To me, that's what we just said, right? Let's look down here at B. This one says it's water and light intensity. Are the bubbles water? The bubbles aren't water. The bubbles are gas in water. So to me, that's not a good answer choice because we aren't even looking at water compared to the light. What about the plant require energy and lower light intensities? Well, we're looking for the energy, but we can't measure the energy. We can't go in there. We don't have a tool that we're gauging each chloroplast, are we? This will probably be the second best answer because we're using the bubbles as a gauge of energy. But the lower light intensity would be down here. This one makes less sense to me. Okay, and back down here, carbon dioxide for higher light intensities. If you don't remember that those bubbles are oxygen because it puts out oxygen versus takes in carbon dioxide, you might be confused. Okay, to me, you're going to pick one that that is measuring the gas and the light. And if you know that that gas that it's putting out is oxygen, you're going to pick A. Does that make sense? You have to know that the gas that is putting it out through photosynthesis is oxygen. Plants don't put out carbon dioxide. They take in carbon dioxide. So these oxygen bubbles that they're releasing, Jesse, we have to know that those gas bubbles are oxygen. Kind of a tricky question, right? I know, we're running late. We will finish this question next week. These application things are what we have to break down. Okay? We've got to be able to break these questions down. In this case, hey, Mila, can you be quiet for a minute, please? In this case, we have to connect the products and reactants of photosynthesis. We have to know what the plant takes in and what the plant puts out. If we don't have that in our mind going into this question, it makes it really hard to answer it correctly. You guys are rock stars. Don't get bound by this. I know it's tricky, but we're going to work through these and work through these so that you're ready when we get there. And you're like, all right, I know what they're doing to my mind, but I'm going to break this down and I've got it. Okay. I will see you later. Have a great day. Keep practicing, everyone. You've got this. Bye.